We'll see how long this lasts. I don't think this is gonna last too long. One, because my internet speed. Yeah, I'm already dropping every frame. Let's drop this bit rate. Well, we'll see. But yeah, the bit rate is. Can I change the bit rate live? I don't know if I can. Oh, I think I can. Is it gonna drop? There we go. Am I live? Let's do some biology homework. I'm on a boat.
Flagella, singular flagellum, and cilia, singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Was that it? The cilia and flagella shows the microtubules. Gap junctions happened. Small channels are formed. Okay, so these are called gap junctions. Membrane proteins that create a watertight seal between. It's like I'm on a boat. How much of this am I gonna do? It's my life. I'm on a boat. Let's see, am I dropping hella frames now? I feel like it's probably gonna end, and if it ends, I'm just gonna like let it end. But we're gonna keep this going as long as we can. I got electricity. I don't have unlimited hotspot data usage, but we'll see how much I get. Tight junctions. Okay, that's tight junctions are what are membrane proteins that create a watertight seal between cells, and that's animals. Did I just closed the wrong one? Of course, I closed the wrong one. Okay, hold on. I'm on a boat. Connections between cells. Alright, let's see. Oh, I lost all my stuff. Okay, that's good. I gotta do it again. So the plasma desmata or this. That one I remember from last quarter. The desmosomes. That was animal? Okay, I would have gotten that wrong. Desmosomes. Gap junctions. Tight junctions. A polysaccharide that is used to synthesize cell walls and protect. That's definitely cellulose. Long fibers in the ECM, that's collagen. I think collagen is like the most abundant protein in the human body. I'm pretty sure that's true. Um, okay, plant. The cellulose, that's obviously plant. And then ECM, or collagen. That's got to be right. It's my life. Kind of surprised how well this is working. I just wish I had Starlink. Starlink would be the way to do this, bro.
Animal cells in multicellular tissues are usually joined by tight junctions. Desmosomes, also called anchoring junctions, and gap junctions, also called communicating junctions. Plant cells are often joined by plasmodesmata. Each type of junction has specialized functions, such as preventing fluid leakage between membranes or allowing the free passage of small molecules and ions between adjacent cells. Tight junctions, which bind cells together, are found predominantly in epithelial tissue, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells. Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins, such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton. In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow the passage of water and small molecules. Gap junctions help coordinate the activities of adjacent cells. For example, a hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions. Plant cells, unlike animal cells, are surrounded by thick walls that form rigid tissues. They do not need the junctions found in animal tissues. But some higher plant cells are interconnected by plasmodesmata, tube-like structures that penetrate through cell walls and form fluid connections between adjacent cells. Like gap junctions in animal cells, plasmodesmata allow the free exchange of small molecules and help coordinate the activities of neighboring cells.
Lido. Wait, no. Site. Oh yeah, Mido means thread. I actually knew that. I knew that. Trust me, chat. I knew that. I knew that because that's a weird one. Not a regular one. It's my life. Loki, I'm surprised this is working so well. High key, low key are actually synonyms. Phil. What is Phil? I don't remember Phil. Hyper is above and greater than. This is E. Sim. Sim, I think, is actually the same as sin, which is. Micro is small. Oh, wait. Hypo is below. Plasm. Plasm is like plasma is formed. Fill is leaf and sim is same. I love etymology, bro. I love etymology. Biology, if you like etymology, you gotta do biology for sure. No other science has as much etymology in it.
Geometric relationships between surface area and volume. As organisms get larger in size, they do so by making more cells instead of just increasing the size of a few cells. This figure explains why. Each cube in the figure represents one cell. Cells are not perfect cubes, but making them that shape for our example keeps the math simple. An important relationship for cell size is the ratio of its surface area to its volume. This is because the surface of the cell is the plasma membrane, which is a selective boundary for everything that enters or exits the cell. There's a limit to how fast things can cross the membrane in a given area, so if the surface area is too small compared to the volume, the cell might not get enough of what it needs, such as oxygen, to support the whole cell interior and stay alive. Let's look at what happens to the surface area to volume ratio as the overall size of a cell changes. The first example is a small cell that measures one arbitrary unit on each side. The total surface area is calculated by determining the surface area of one cell, then multiplying by the number of cells. This small cell has a height of 1 unit and a width of 1 unit, so the area of one side is 1 unit times 1 unit, which equals 1 unit squared. There are 6 sides, each with the same area, so the surface area of the small cell is 1 unit squared times 6 equals 6 units squared. Here, there's only one cell, so we multiply by 1 to get the total surface area of 6 units squared. To calculate the total volume, you first need to calculate the volume of one cell by multiplying its height times its width times its length, so you end up with units cubed. The small cell has a volume of 1 unit times 1 unit times 1 unit equals 1 unit cubed, and since there's only one cell, its total volume is 1 unit cubed. Once the surface area and volume are calculated, you can determine the surface area to volume ratio by dividing the surface area by the volume. For this small single cell, the ratio is 6 divided by 1 equals 6. What happens to the surface area to volume ratio if the cell increases in size to 5 units on each side? A similar calculation shows that the surface area of this larger cell is 150 units squared, and its volume is 125 units cubed. For the larger cell, the surface area to volume ratio is 1.2, which is much smaller than for the small cell and might be too small to support its transport needs. Because the surface area increases as a square of the dimension, and the volume increases.
Suppose you discovered a new multicellular organism that is shaped like a cube. The organism measures 3 cells tall, 3 cells wide, and 3 cells deep. Measure one cubed cell to be one cubed cell to be 2 arbitrary units per side. Can you calculate the surface area to volume ratio? Okay, so the organism's surface area to volume. All right, so there's 27 cells. 4 times 6 is 24. 24 times. All right, we gotta do this. What? Oh. Okay. 3 to the 3 cells times 2 squared times 6. That's the surface area. But the volume is just going to be 6 by 6 by 6, 216. Here's the best description of the cell cytoskeleton. The cell cytoskeleton is a dynamic network of fibers that can be quickly dismantled and reassembled to change the cell shape. The typical diameter of a eukaryotic cell is 10 times the diameter of typical. I'm really surprised this has been going on for as long as it has. 20% drop frame is actually totally fine with me. I'm actually probably, just because I want to keep this going as long as possible, I'm probably going to go. I'm going to drop this even lower. Who cares about the bitrate, bro? I'm on a. Oh! Alright, what's next? Oh yeah, I gotta do this thing. Okay, so this one's done. This one I need to do. Alright, this one's gonna be on biotechnology. Okay, biotechnology. Okay. Enzymes are used to cut and paste DNA. To create a molecule of recombinant DNA, which of the following is cut with a restriction enzyme? Seven minutes.
Hey, this is Eric Simon from New England College in Henniker, New Hampshire. In this Visualizing the Concept video, we'll walk through the process of creating recombinant DNA. Let's get started. Many genetic engineering experiments involve combining DNA from more than one source, often from different species. For example, a human gene encoding for an important protein may be inserted into a bacterium. Such a modified bacterium will then reproduce and express the human protein, which can then be harvested in large quantities, perhaps by a pharmaceutical company to produce a new drug. In another example, an insect gene might be placed into a plant in order to improve that plant's ability to resist cold weather. To create recombinant DNA, scientists must cut pieces of DNA from multiple sources and then paste them together into a single recombinant DNA molecule. In this animation, we're going to focus on the actual cutting and pasting of DNA to see how a typical genetic engineering project is undertaken. To help visualize this process, let's use household materials. Imagine that you have a necklace made from blue ribbon, and you wish to insert a bit of red ribbon into it.
To do this, you would snip out a piece of the red ribbon by making 2 cuts. Then open up the blue necklace by making 1 cut. You could then insert the new ribbon and seal the gaps. You now have a recombinant necklace that contains pieces from 2 different ribbons combined together. To accomplish a similar process with DNA, genetic engineers often start with plasmids, small circular pieces.
Which of the following statements is correct? If a restriction enzyme is cut... What? If a restriction enzyme is cut at its site, the result is one or more restriction fragments. What the hell?
Pieces of DNA found within many bacteria, such as this E. coli. Once a researcher has removed a plasmid from a bacterium, it can be manipulated in the laboratory. In this experiment, the circular plasmid needs to be cut open. This is accomplished using a restriction enzyme, a protein that cuts DNA at one specific sequence of bases. The restriction enzyme shown here, for example, is called EcoR1. It cuts DNA at one specific sequence of bases called the restriction site. For example, the restriction enzyme EcoR1 will only cut DNA at the sequence GAA-TTC. When this restriction enzyme is added to the plasmid DNA, it will cut the DNA at every location that contains the sequence, and no places that don't. The specific restriction enzyme used in a genetic engineering project varies from experiment to experiment, but an engineer will usually choose a restriction enzyme that will cut the bacterial plasmid at just one location, opening up the circle. If a plasmid contains 2 restriction sites for a particular restriction enzyme, how many separate pieces of DNA will result? The result of cutting a circular plasmid twice will be 2 linear pieces of DNA. Imagine snipping a rubber band twice. You'll be left with 2 pieces. Once the bacterial plasmid is snipped open, it's ready to have another piece of DNA inserted into it. Here you can see a human cell that contains a target gene. Once the DNA is extracted from the cell, the same restriction enzyme can be used to cut it up. Because the human chromosome is so big, it will probably be cut into several pieces, one of which contains our target gene, shown in red. If a linear piece of DNA contains 2 restriction sites for a particular restriction enzyme, how many separate pieces of DNA will result?
If a linear piece of DNA contains 2 restriction sites for a particular... Oh, it's 3.
In this case, since the starting DNA is linear, 2 cuts will result in 3 linear pieces of DNA. Imagine cutting a strip of paper twice. You'd be left holding 3 smaller strips of paper. Now that we have our 2 desired segments of DNA, 1 bacterial plasmid, and 1 human gene, let's see how they're pasted together. The key is that the restriction enzyme used did not cut across the DNA double helix evenly. Instead, it left staggered cuts called sticky ends. Each sticky end is a single-stranded set of unpaired DNA bases. The bases in the single-stranded regions are no longer hydrogen bonded to their matching bases on the other strand. These unpaired bases are now available to pair up with a complementary set of bases on another sticky end. Notice that the DNA base sequences of the sticky ends on the bacterial plasmid DNA and the human DNA match up. The key to creating recombinant DNA is to use the same restriction enzyme to cut both the plasmid DNA and the target DNA. Because the same enzyme was used, the same sequences were cut in the same way. As a consequence, the unpaired base sequences are complementary to one another, and the sticky end of one piece of DNA will match up with the sticky end from another. The hydrogen bonds that form between the sticky ends of the 2 pieces of DNA temporarily hold the foreign DNA in the middle of the plasmid. To permanently paste these separate pieces of DNA, the engineer can add a DNA pasting enzyme called DNA ligase. This enzyme builds new covalent bonds, sealing up the separate pieces of DNA into a single recombinant piece of DNA. The goal of creating recombinant DNA has now been achieved. Once created, the recombinant plasmid can be inserted into a bacterium. The recombinant plasmid will multiply to produce a colony of identical bacteria, each of which carries the foreign gene in a recombinant plasmid. As it goes about its life cycle, a recombinant bacterium will treat the foreign DNA as if it were its own. This means that the foreign DNA will be transcribed into an RNA molecule and then translated into a protein. Perhaps the protein is a pharmaceutical product that can serve as a drug. Right now, in the US Midwest, there are large tanks of recombinant bacteria carrying the gene for human insulin. The bacteria transcribe and translate the gene to produce human insulin protein, which can then be extracted and purified. This human insulin is then packaged and sold to millions of diabetics, for whom this drug, made from recombinant DNA, is a life-saving medication.
That rules. So wait, what is the question then? I don't even get this question, dude. What is this question? That's probably the answer. Check each of the true statements with the protein involved. I think it might be just those two. Oh, did I not click that one? I don't know why I didn't click that one.
Often, it is important to determine the length of each DNA fragment on an electrophoresis. This may be easily done if a marker DNA sample is run alongside the experimental samples on the gel. The marker DNA sample contains a number of DNA molecules, each with a known molecular size. Molecular size is measured in terms of number of base pairs in a DNA fragment. Our marker DNA includes 8 DNA fragments that have 23,130, 9,416, 6,557, 4,361, 2,322, 2,027, 564, and 125 base pairs. The 125 base pair fragment is often not seen in experiments either because it is too faint or because it runs off the gel. This video shows an example of a gel with this DNA marker and some unknown DNA samples, DNA 1 and DNA 2. To determine sizes of unknown DNA fragments, a calibration curve is first made of the known marker DNA data. For each fragment, the y-axis shows the log of the number of base pairs, BP, and the x-axis shows the distance that fragment traveled in millimeters, mm. A new calibration curve must be made for each gel because the exact migration distances of marker DNA fragments vary from experiment to experiment. Next, the migration distances for the unknown DNA bands are measured, and the sizes of the DNA fragments are read from the calibration curve. Now, let us consider the orange band as a fragment in DNA and determine its length. A line will be drawn from the distance the fragment traveled on the x-axis to the calibration curve. At the point that it intersects the calibration curve, another line will be drawn to the value for the length in base pairs of the fragment. In DNA 2, there are 3 fragments. See the length of the first band. It is 4,796 base pairs. Length of the second band is 1,405 base pairs. Length of the third band is 1,268 base pairs.
The ability of restriction enzymes to cut DNA at specific sites makes DNA cloning possible. The diagram below shows a section of human DNA that contains an imaginary gene for video game proficiency. That's pretty good. Alright. Which enzymes produce DNA that contains the entire HIND for sure, BAM for sure, BAM BAM, BAM BAM. Inserting the VGP vector into a cloning gene into a cloning vector. Oh, that's a big one. In the second step of the cloning procedure, DNA fragments produced by a restriction enzyme are inserted into a cloning vector that has been cut by the same enzyme. The vector used in this tutorial is a plasmid. To clone the gene into the plasmid below, a restriction enzyme must do all of the following. Cut the human... Actually, wait, I want to look at the stream. How good is the quality? Is the quality any good? It actually seems like totally fine. To clone the gene into the plasmid, a restriction enzyme must do all the following. It must cut the human DNA on both sides of the VGP gene, but not inside. Cut the plasmid without cutting inside the ampicillin. That makes sense. Produce sticky ends. Okay. Which enzyme would cut the human DNA? Which enzyme? Wow, we're trying to engineer. Dude, the whole premise of this homework question is hilarious. We're trying to make a gene. We're trying to make a drug that we can give to people and sell that's going to make them good at video games. That's the whole point of this homework problem. I'm glad we're learning to do important things. On both sides, but not inside. Isn't that the same thing? Well, actually, it's going to be... It said on both sides, but it didn't say anything about the sticky ends. So it's actually going to be Hey 3, BAM, HI, and HIND. So it's going to be everything but EcoRI. BAM, Hey, and HIND. Which enzymes would cut the plasmid? Is it? Oh, this is AMPR. Which enzymes would cut the plasmid without disrupting the function of the AMPR? Hey, BAM, and ECO. Hey, BAM, ECO. Which enzymes would produce sticky ends when cutting both the human DNA and the plasmid? BAM, and HIND, and ECO. BAM, HIND, and ECO. BAM, HIND, and ECO. So it's gonna be BAM, HIND, BAM, HIND, and ECO. Which one satisfies all the requirements? It's gotta be BAM. Did I do that right? Transformation is selection of recombinant bacteria. In the third step of the cloning procedure, all the plasmids that were created in the third step are incubated with bacteria. The bacteria do not have an AMPR gene on their own chromosome. As shown below, at least 4 classes of results. So we put in a off-target one. We didn't put anything in. Somehow we didn't get the plasmid, and then we got both. Will only grow in a medium without ampicillin. Will grow only in medium containing ampicillin. What? Screening the plasmid library. The final step of the cloning procedure is to screen the plasmid library for clones that carry the VGP gene. To accomplish this screening, researchers synthesize a single-stranded DNA probe using the VGP mRNA as a template. Probe? What does this actually mean? Wait, am I really gonna have to... What? What the hell? Okay, wait, so it's going 5-3. So, I don't know really what the point of the probe is. Did it say single-stranded? So this is going 5-3, then shouldn't it just be a... Wait, hold on, let's do this. The probe is a DNA transcript of the VGP mRNA, which itself... Oh, do I have to do a double flip? Which is itself a transcript of the VGP gene's template strand. So presumably this is going to be the template strand because we're going to add 5-3. I mean, we could also go this way, though. So couldn't you also put T here? Okay, find the template, transcribe the template, and do a strand of mRNA. Whoa, this problem is kind of crazy actually. So okay, let's just start. We're gonna make the this would really be U. So I'm not doing this for real, but if the bottom is the template, then this is going to be the mRNA, which kind of makes sense because we have, I think ATG is like methionine, which is the start codon. But then we're going to do this again. So then we're going to make a DNA strand complementary to this. So I'm pretty sure then this is going to be like, this is going to be A, which is this top thing. But then we're going to do it again. So I'm pretty sure that this is just going to be the template strand. So it's going to be T, A, C, like that. Wait. Oh, okay, yeah. I mean, that's fine. It's still ATG. It's just the mRNA is ATG. Okay, that I don't need to do. All right. So I'm pretty sure that I have it here. The TAC T T A. You guys see that these have been like like who made this? Like look at how they're the height is different and then the horizontal spacing is totally inconsistent. Who made this? It's like someone had to make. They must have like a tool. Like Pearson must have a tool that allows teachers to like make their own assignments or something like that. I don't know what the deal is. AAG. Okay, G T. Can we do this faster? C GTC ACA. I think that's right. If you're going through. Okay, wait, hold on. We make an mRNA template. Oh, okay, wait. We make an mRNA template. This is the template then. Wait. No. Hold on. Hold on. We gotta think about this. I don't know why I didn't think about this. Okay, so which is the template strand? The template strand is complementary to the mRNA, which begins with the start codon. Okay, so this is clearly the template. So then we're going to have a 5' going 3' transcript. But then we're going to go this way. Oh, so I got to flip all this shit around. Damn it. Yeah, I messed that up. Okay. ECA C TCACA. Hold on, I gotta. Can I delete? If I click this. Oh shit. What? Select this, go here. Oh, because it's a different... It's... Bro, what? How do I move it out of here? Oh wait, clear all. Okay. T, C, A, C, A, CTG, C, T, C, A, C, T, C, A, C, A, C, T, G, G, A, A, T, T, C, A, T.
Near the base of the brain, a pea-sized structure called the pituitary gland secretes a variety of hormones into the bloodstream. Some of the pituitary cells secrete growth hormone, which stimulates growth during childhood. Children who produce little or no growth hormone grow more slowly than average, reach puberty 2 to 10 years later than average, and are short in stature as adults, a condition called pituitary dwarfism. However, if these children are treated with injections of human growth hormone, they can grow to normal height. How did scientists produce a supply of human growth hormone? Essentially, they mimicked in the laboratory what certain pituitary cells do in the brain. In these cells, the growth hormone gene, called GH1, is transcribed into mRNA, and the mRNA is then translated into the growth hormone protein. The laboratory project to produce human growth hormone had three main parts. First, the scientists created a cDNA library, which is a collection of DNA sequences from the genes expressed by a sample of cells. Second, they screened the library to find the growth hormone gene. Finally, after finding the gene, they placed it in bacteria. The bacteria transcribed and translated the gene, thereby acting as microscopic growth hormone factories. Scientists began the hunt for the growth hormone gene by extracting messenger RNA molecules from pituitary tissue. In addition to hundreds of other types of mRNA molecules, the pituitary gland produces mRNAs transcribed by the growth hormone gene. The researchers constructed a cDNA library by converting this mixture of mRNAs into a form that could be manipulated in the laboratory. To make a cDNA library, an enzyme called reverse transcriptase copies the mRNA into a strand of complementary DNA, or cDNA. Next, the RNA is destroyed by chemical treatment, and another enzyme, DNA polymerase, copies the remaining DNA, making a double-stranded cDNA molecule. These cDNA molecules contain the same nucleotide sequences as the original messenger RNAs. The next step in constructing the library is to insert these cDNA molecules into loops of DNA called plasmids. This procedure allows the DNA to be replicated in the laboratory. The plasmid contains a recognition site where a specific restriction endonuclease can cut it. These recognition sites are added to the cDNAs and sealed in place by the DNA ligase enzyme. A specific restriction endonuclease then cuts all of the recognition sites, leaving overhanging sticky ends on the DNA. The ends of the DNA fragments are called sticky because the single-stranded bases on one fragment are complementary to the single-stranded bases on the other fragment. As a result, the ends tend to pair up and form hydrogen bonds with each other. The fragments are permanently sealed together by DNA ligase. The plasmids with the cDNAs are recombinant plasmids. They have different colors added here to show that they contain the codes from different genes. The recombinant plasmids were inserted into E. coli bacteria, forming the cDNA library. After the bacteria replicated the plasmids, the investigators could begin to analyze the cDNAs. In this cDNA library, only one or a few of the recombinant plasmids contain the coding sequence for the growth hormone protein. To find the bacterium with this particular cDNA, the researchers performed a multi-step procedure. In the first step, they spread the cells over a plate and allowed them to divide to form colonies. Any one colony contains millions of the same type of cell with the same recombinant plasmid. In the next step, the researchers laid a filter onto the colonies and then pulled it off. Some of the cells remained on the plate, and the rest stuck to the filter in the same positions as they were on the plate. The filter then contained a replica of all of the colonies that were on the plate. Next, the investigators chemically treated the filter to release the DNA from the cells and make it single-stranded. As a result of this treatment, the DNA was stuck permanently to the filter. To find the colony with the growth hormone gene, the researchers needed to construct a probe, a short piece of DNA that would match and bind to part of the gene sequence. Yet without having found...
Hold on, I low-key lost focus. There's a bunch of freaking seagulls around me. Whoa. Did they talk about that yet? That's... Oh, did I close it? Why did I close it? I thought I...
Near the base of the brain, a pea-sized structure called the pituitary gland can cut it. These recognition sites are added to find the colony with the growth. The activity was what feature the encoder sequence odor and transcribe.
Where the hell do they talk about
Eight parts.
cadavers, bro? What? Oh. Yeah, let's watch it. Because,
First, ignition sites, leaving overhanging sticky ends on the DNA. The ends of the DNA fragments are called sticky because the single-stranded bases on one fragment are complementary to the single-stranded bases on the other fragment. As a result, the ends tend to pair up and form hydrogen bonds with each other. The fragments are permanently sealed together by DNA ligase. The plasmids with the cDNAs are recombinant plasmids. They have different colors added here to show that they contain the codes from different genes. The recombinant plasmids were inserted into E. coli bacteria, forming the cDNA library. After the bacteria replicated the plasmids, the investigators could begin to analyze the cDNAs. In this cDNA library, only one or a few of the recombinant plasmids contain the coding sequence for the growth hormone protein. To find the bacterium with this particular cDNA, the researchers performed a multi-step procedure. In the first step, they spread the cells over a plate and allowed them to divide to form colonies. Any one colony contains millions of the same type of cell with the same recombinant plasmid. In the next step, the researchers laid a filter onto the colonies and then pulled it off. Some of the cells remained on the plate, and the rest stuck to the filter in the same positions as they were on the plate. The filter then contained a replica of all of the colonies that were on the plate. Next, the investigators chemically treated the filter to release the DNA from the cells and make it single-stranded. As a result of this treatment, the DNA was stuck permanently to the filter. To find the colony with the growth hormone gene, the researchers needed to construct a probe, a short piece of DNA that would match and bind to part of the gene sequence. Yet without having found the gene, how would they know what nucleotide sequence to use for the probe? To solve this problem, the investigators analyzed part of the protein's amino acid sequence, which was already known, and then worked backward to deduce an approximate nucleotide sequence for the probe. To track the probe during the experiment, they labeled it with a radioactive atom. The filter was incubated with many copies of the probe, which spread over the filter and into the regions containing the DNA. Note here that the probe binds selectively to the DNA in one colony and not in another. It binds only to the DNA in colonies that contain the growth hormone DNA, because the bases in the probe are complementary to the bases in the growth hormone DNA. The excess radioactivity was washed off the filters, leaving radioactivity only in the location containing the gene for growth hormone. The researchers lined up the filter and the plate to find the matching colony. Using this procedure, the investigators identified the colony of cells that contained the growth hormone cDNA. When the researchers found the colony with the growth hormone cDNA, they prepared it for expression in bacteria. They removed the cDNA from its original plasmids inserted back into E. coli cells. The cells recognized the promoter and transcribed the cDNA into mRNA. The mRNA was then translated into the growth hormone protein. The human growth hormone that accumulated in the bacterial cells as they grew could be isolated and purified. Bacterial cells containing the human growth hormone gene are now grown in huge quantities.
It's getting cold. I go downstairs. Oh, wait, but actually, let me do this. Yeah. So which of the following steps would be performed last when screening a cDNA library? How can large quantities be produced? The term sticky ends refers to the overhanging.
Overview. The polymerase chain reaction, or PCR, is...
Fuck, I know this. Hmm. Let's see.
is a powerful method for obtaining millions of duplicates of a DNA fragment from just one original copy. The reaction begins with a solution containing the DNA from a cell. The goal is to amplify a specific region, the target DNA. The solution contains Taq polymerase. Taq is a type of heat-stable DNA polymerase derived from a species of bacteria living in hot springs. Because Taq polymerase continues to function normally at high temperatures, using it allows researchers to separate the DNA strands without destroying the polymerase. The solution also contains primers, short single-stranded DNA molecules, and plenty of deoxynucleoside triphosphates, or DNTPs, which are not shown at this scale. The PCR cycle. Let's examine the basic PCR cycle, which is typically repeated about 35 times. Temperature changes are automated by PCR machines. In the first step, denaturation, the solution is first heated to nearly boiling, 95 degrees Celsius. The heat breaks the hydrogen bonds between the two DNA strands and allows them to separate. Despite the intense heat, the Taq polymerase remains active. In the second step, primer annealing, the temperature is dropped to around 60 degrees Celsius. The exact temperature depends on the length and base composition of the primers. At this relatively low temperature, the primers can form hydrogen bonds with the single-stranded DNA. Two primer types are created, each one complementary in sequence to one of the two ends of the target DNA. To make the primers, the sequences at the ends of the target DNA must be known. In the third and final step, extension, the temperature is increased to 72 degrees Celsius. This is the optimal temperature at which Taq polymerase functions. The primers are essential in this process.
Two to the four. Okay. Off by one, add out. Which step are nucleotides used? Extension. Which step in the PCR cycle do primers form bonds? Annealing. Right. Wait, did I miss something? Wait, don't I get to try again? Oh, I gotta try again. Hold on. Oh my god. When they go so, like, the annealing step, like, why would they do that? Okay. Methylated. I gotta do this again. I gotta do this whole thing again. I'm not losing points, bro. It's not in annealing. It's in denaturation. Okay. Oh, wait. Oh, I did on the wrong one. Bro. They did it. They flipped the order on me. Okay, wait. I think I might get one more try again. I don't know. Nucleotides extension. Primers, annealing. Oh, good, I get to try again. Okay, this is the last try again. I can't fuck this up. Whether the gene is methylated. Amplify a fragment of DNA. Comparison gives insight. That is true. Okay. Wow, that was a pain in the butt. Extension. Okay, finally we did it. Oh, yeah. Am I still alive? Man, I am surprised. This thing has gone on so well. Holy fuck. Alright. The CRISPR-Cas system functions as a form of adaptive immunity in prokaryotes to defend against pathogens. CRISPR stands for clustered, regularly interspersed short palindromic repeats, which are encoded in the bacterial genome and are heritable. After a viral infection, a bacteria contain a molecular memory in the CRISPR locus of viral DNA. In the CRISPR locus of viral DNA fragments to help protect itself against future infections by the same virus. Transcription of the CRISPR locus in the bacterial genome produces cRNAs that are complementary to specific sequences of viral DNA. If previously encountered virus infects a bacterium again, the cRNA and a non-coding called a transactivating tracer RNA. What does cRNA stand for? CRISPR RNA? Probably CRISPR RNA. Help guide an endonuclease called Cas to the target DNA in the viral genome. Upon reaching its target, domains in the Cas endonuclease act like molecular scissors and cut the viral DNA. After the discovery of the role of CRISPR-Cas in the microbe community, scientists realized that the system could be re-engineered to target any DNA sequence of interest and edit the genome of a living cell, including eukaryotic cells. By designing crinas that are complementary sequence of interest in the target genome, scientists can essentially build customized scissors to cut in specific places, enabling more precise editing than was ever possible before. Furthermore, fusing the crinas and the tricernas, the crinas and the tricernas sequences in a single RNA molecule called a single guide RNA, siRNAs. The machinery for cutting DNA is simplified to just two components, the siRNA and the Cas. Sometimes siRNA, also called guide RNA, Although this tutorial will use the term Sigourney. Well, different prokaryotes use several different types of Cases, and scientists use Cas9. Cas9 is a single protein, unlike some other ones. Researchers select genomic targets to edit, then engineer specific sequence for the Sigourney to be complementary to the target. Each genomic target must have an adjacent PAM, protospacer adjacent motif, a 5' 3' 5 NGG3 sequence, which is common in the genome. Why is that? Sigourney helps Cas9 to target by binding to the target DNA via complementary base pairing. The Cas9 makes double-stranded DNA. Cas9 makes double-stranded DNA breaks at the target location. After the breaks, hold on. After the breaks are made, the breaks are repaired through endogenous mechanisms. Endogenous. Depending on the break quality like that. This diagram is confusing me. Okay, let's read the things. So that's this one. Whoa. There's so much writing here, man. When DNA breaks are detected in a cell, whether induced by Cas9 or otherwise, the endogenous double-strand break repair machinery of the cell attempts to repair the breaks. Double-stranded breaks in the genome may be repaired by either non-homologous end joining or by homologous recombination called homology-directed repair. NEDG simply involves non-homologous end joining simply involves the ligation of broken DNA fragments. Homology-directed repair uses an undamaged homologous chromosome or sister chromatid as a template to repair a broken chromosome. Cells may use Non-homologous. Bro, can we just get on with this? I have other shit to do. Alright, I guess I have to understand this. So, what this is all about. Okay. Indels. Oh, wait, is my phone? Oh, how did I do? This is not going to be good. Hopefully it's a 9 out of 10. Could be even lower. 8.4. Bro, that one's the one getting dropped. My grade probably just dropped in this class. Oh, I still have a 97. I'm good. Hey. It actually reconnects so quickly. It's great. Um. All right, let's read this thing. Uh, they differ in how error prone they are. Can exploit that to cheat their genome editing goals. For instance, non-homologous is error prone and often results in small insertions or deletions. They can, cause of frame shift or whatever. If the goal of the genome editing is to disrupt the gene and create A null allele. Scientists can use the to make cuts, let the endogenous repair mechanism do its job without further action. The HDR mechanism may repair Cas9-induced double-stranded breaks correctly and incorrectly in others. Hmm. I have no idea. I have no idea what the answer to that is. Holy cow, this one's crazy. Well, CRISPR originated as a mechanism for prokaryotes to defend against. I gotta hurry up. If dCas9 is attached to the catalytic domain of an epigenetic modifier enzyme, That's cool. Researchers want to make an endonuclease that binds to target DNA, but does not cut. Okay, the scientist first obtains all of the mRNA connected in the cells of interest. Using the mRNA as templates, a reaction produces single-stranded DNA that has complementary sequence. So that's like reverse transcriptase or something. This DNA, called cDNA, is fluorescently labeled and added to the glass slide. If a cDNA molecule contains a sequence complementary, it binds. So now you can see which mRNA is in. Gene K is highly expressed in. No. For some genes, there's variation in. If you were given a microarray data from the cells of a new patient belonging to the group 1 group. The patients in group 1 have acute lymphoblastic leukemia, ALL. In ALL cancer cells arise from a lymphoid precursor from cells that normally have specific types of white blood cells. Normally forms acute myeloid leukemia, AML. Cancer cells arise from a myeloid red blood cell. Okay. F high, P high, G group 1, B high, H high, S high, S low, T high, S low, U high. Probably. Oh wait, no, ALL. B high, Q high. Probably AML. But okay, wait, hold on. Auxin 9, patients with high expression of HOXA9 experience treatment failure. Scientists hypothesize that low HOXA9 expression may make a patient more likely to survive. Patient 1 versus 10. What are the genes? X and G. One has high X. Okay, 6 versus 16. 6, low X, low G. And 16. 16X low, but really high G. G was the Hoxa. I'm pretty sure G is the Hoxa. So Hoxa, they have high Hoxa. So what they're wondering is people with high Hoxa, who's going to be better off? Patient 6. Yeah, patient 3, low X, high G versus 10. Low X, high G, low X, low G. So this is, do people with high G patient 10, I think. Let's just see if I get it right. If I don't, I'm just going to go chat GPT it. Okay, good. Okay, gene edited supercells make progress in fight against sickle cell. When was this? 2019? So what did they do? How did they do it? What did they do? Whoa. Fetal hemoglobin is a protein that is normally produced only by fetuses and newborn babies for a short time after birth. So scientists use CRISPR to edit a gene in bone marrow. Scientists use CRISPR to edit a gene in bone marrow cells that had been removed from Gray's body. The edited cells were infused. Can I just ask questions about this? Okay, I just want to ask what. So what happened? Use Kurtz to edit a regulatory DNA enhancer. Oh. So they knocked down. Yeah, so they knocked down. So they. Knocked down a repressor on the. Okay. Do 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 do. You are a doctor that specifies. Do 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 do. Cruising down the street in my 6'4". It will have no effect on the chance of people having sickle cell disease. It will greatly reduce the chance of people having sickle cell. You hope to study a gene that codes for a neurotransmitter protein produced in human brain cells. You know the amino acid sequence of the protein. Arrange the following techniques to demonstrate how you might identify and isolate the neurotransmitter gene. Um, mm, 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 mm. Bum, 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 bum. Bro, you gotta get a move on. Mm, 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 mm. Bro, I ain't reading all that. Actually, I will. Water in Yellowstone 70C. Biologists assume that no species of organism could live in water, but so they're surprised thermophiles. You learned in this chapter how an enzyme from one species, TAC, made feasible one of the most important DNA-based. What the hell? Is that an image? What is that? You can't select that? What was the enzyme? What was the value? TAC was isolated. No, not restriction. TAC polymerase. Can you think of other reasons why enzymes from this bacterium might also be valuable? Enzymes from thermophiles that researchers. I feel like this one could be true too. Wait, other enzymes lose. I feel like that's not true. Let's try its answer. But I don't think that's right. Yeah, that's so wrong. Alright, I'm just gonna guess mine, which is neither of these. That's what I thought. Bruh. Which of the following is a concern expressed by opponents of GM? I don't know. I don't actually know about that. A researcher is preparing to insert a human gene of interest into bacterial plasmid in order to clone the human gene. She's genetically engineered the plasmid to carry AMP-R, which confers resistance to ampicillin. She will include ampicillin in the plating media when she grows the recombinant bacteria. Why has she engineered the plasmid? I think that's actually. Is that true? What's up with the blue thing? I think lac. No. What is it? I forget what it is, but there is actually something about the like. What was it? Was it just lac or something? But there was something. Okay, finally. So now I have to do like some IR spectroscopy thing. Which I don't know anything about that. Okay, but I'll be back. I'm gonna go take a leak. A leak? Wait, but actually, can I take a leak? No one's gonna come here. It's gonna be fine. Okay, I'll be back. I'm back. I'm back, bruh. Grandma smells like shit. Grandma smells like shit. Grandma smells like shit. Granny smells like shit. Anyways. So wait, so I'm actually good on that. Granny smells like shit. Granny smells like shit. Alright, I probably got 3 disconnect notifications or something. Granny smells like shit. Grandma smells like shit. Grandma smells like shit. Granny smells like shit. It's such a disrespectful song, bro. Like, who come up with that? Alright, wait, so now I have to do this. Okay, so what's going on here? Please circle the compound which best fits the IR spectra shown. Granny smells like shit. Grandma smells like shit. Grandma smells like shit. Oh shit. Oh no. It's not good. All this stuff here is old. Alright. Granny smells like shit. Granny smells like shit. Granny smells like shit. Okay, wait. So if we just like looked at the table, I actually don't remember how to do any of this shit. All right, here we go. All right, here we go. We're just gonna apply this algorithm. I think it's funny that like chemistry is like people don't know it, but chemistry is full of algorithms. What is it called? The Con rules RS OChem. The CIP, that's what it is. The Kohn-Ingold Prelog rules. That's an algorithm. Determining oxidation state, that's an algorithm. All of the like little things, it's like flowcharts and algorithms. Alright, so let's just do this thing. This should be easy. Okay, so wait. Carbonyl, C double bond O. This is a okay, so wait. Let me get my notebook. Granny smells like shit. Granny smells like shit. Grandma smells like shit. Grandma smells like shit. Granny smells like shit. R.I.P. my granny, she got hit by a bazooka. R.I.P. my granny, she got hit by a bazooka. Alright. Alright, so alright, so what is this? This is like 2,2-dimethyl 4 2 oh okay, it's gonna be like 2,2,4 trimethyl pentane or something like that. Yeah, 2,2,4 trimethyl pentane. Okay, so let's just do this. We'll go 1 2 3 4 5. Okay, 1 equals 2,2,4 trimeth pent. Oh yeah, nomenclature. There's a ton of like algorithmic shit in that. Okay, 2 is. It's probably. Oh, these ones are interesting because longest carbon chain. Let me think about that. Alright, so let's draw a benzene. So it's like this. We got benzene. We got 1, 2, 3. But then we have double bond O, OH. That's what we have. So, I forget how to name things with rings. Because I think if the ring is bigger than the longest chain, don't you name it, like, based on that?
R.I.P. my granny, she got hit by a bazooka.
Benzene ring CH2, CH2, COH. Okay, so 1, 2, 3, 4. The highest priority group is a carboxylic acid. Okay, so the parent is propanoic acid. Okay, wait, yeah, because the 1, 2, 3. So this thing is propanoic acid. Okay. Okay, so then 1, 2, 3. Okay, then this is gonna be a 3-phenyl. So it's 3-phenyl propanoic acid. Okay, so 3-phenyl propanoic acid. Okay, cool. This one is just 1, 2, 3, 4. 1, 2, 3. Okay, this one is phenyl. You know, one. What do you do with the suffix on the amine? I don't even know how to determine which groups. Honestly, I gotta get so much more into nomenclature. Like, I just scratched the surface of nomenclature. NH2. Okay, what about this one? How do you name this one?
R.I.P. my granny, she got hit by a bazooka.
Oh, they're gonna call it para. 4-methyl-aniline. Okay, so this is really called 4-methyl-aniline. Aniline. Okay, so then what is this one? This is probably going to be the highest. It's but it's not a. I think it's an aldehyde. Aldehyde ends in all. So this is going to be like, ooh, this one's tricky. Honestly, it's going to take me like many, many hours to like really learn these things. So I'm not going to do it like intensely. 2, 3, 4. Here. Double bond O. H. This is like benzene. Aldehyde. Hydroxide. I think, is this the whole worksheet? Bro, this is clutch. This is going to be easy. Okay. Yeah. So the aldehyde is the hydrogen. So it's going to be, so the parent is, interesting, benzaldehyde, which smells like cherries. The OH is meta. Okay, so it's going to be 3-hydroxybenzaldehyde. Okay, cool. Or meta. But I like 3. 3-hydroxy benzaldehyde. I thought though that Granny smells like shit. Granny smells like shit. Grandma smell like shit. Grandma smell like shit. Where's nomenclature? It's right here. Where's aldehyde? Halide, alkene, arene, amine, aldehyde. It ends in all. It's a retained. I don't know. I'm not getting too into it. Let's just keep going. Okay, so this one is going to be... Ooh, this is interesting. 2 benzene rings.
2 benzene rings.
I'll be posted up. Okay, so this one's 3-hydroxybenzaldehyde or meta. Biphenyl. Phenyl. What's systematic name of naphthalene? Bro, that's crazy. At some point, it's like, yeah, with tele-fused rings, the only way to really do it is with the graph. Like, it's one of those things that's going to be really hard to, So it's just called biphenyl. I fucked it up. Fucked it up. I fucked it up. Okay, there we go. This is called biphenyl. 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 Okay. Alright, so now let's just go to the algorithm and let's figure this thing out. Okay, so let's go through 1. So this is 1. Is carbonyl present? No. Okay, 2. If it is, okay, but okay, here we go. Alcohols, phenols have a broad signals for amines, ethers, double bonds, sp hybridized. There's nothing. It's a total alkane. Here we go. 7. It's 7 alkane. Main signals for sp3 are in zone or ch or sp3 hybridized ch bonds are in zone 2, just below 3,000 centimeters inverse. Okay, so let's just look then for basically something that has only shit below 3,000. Let's see, how many do we have? 1, 2, 3, 4, 5. Okay, so it's got to be one of these. Maybe the alkane is the hardest to figure out. No, low-key, it's got to be this. Right? Because what else is going to have... Well, I don't know. Okay, so... Should be... The thing is, how many sp3ch bonds are there? This is going to be 1, 2, 3, H, H, H. This is going to be H, H, H. This is going to be H, H. There's a lot of... So let's see here. CH is going to be peaked like right there at, oh, it's probably this. No, wait, at 3100. This is a little low for that. This is like an aldehyde thing. Oh, wait. Oh no, this is probably the alkane. What's above 1500? We have a peak around 1500. Indicative of a double bond. CC double bond. 13 is like a nitro. 29 though is a CH. What's the difference between... Oh, this is sp2. Okay, this is probably... I would say that this is probably structure... I would say this is probably structure 4. Okay, let's do compound. Okay, now we're doing compound 2. Is carbonyl present? So carbonyl... Is carbonyl present? Check zone 4. Go to step 3. Okay, so we have carbonyl on this. So yes, C double O. Okay, so we have the C double O. So strong absorption in zone for 2000 to 1600. Maybe. Oh, that looks like it, bro. That looks like it. Structure 5. Okay, so now that we've looked at anhydrides, what does that say? I can't see that. Is not an S. Okay, carboxylic acid should have a broad spectrum that zone 1 is.
My granny, she got hit by a bazooka.
All right, so this one, so structure 5 is number 2 is structure 5 because it's got a broad, but just look for phenyl groups. Phenyl to the left of 3000. Double bonds. How much of this do you really need to remember? I feel like someone should use software to not just like lame shit like this. Broad carboxylic acid with the, yeah, bro, this is it. The carboxylic acid gives this wide thing. Okay. Number 3. Number 9. Okay, so what's the third compound? It's this 4-methyl aniline. Okay, so we have no carbonyl. If no carbonyl, go to 3. Okay, we're looking for phenols. Phenols have some broad thing in 36. So wait, so if I go 1, 2, 3, 4, 5. Right now, this 1 is going to 4. 2 is going to 5. Okay. 2 is going to 5. Now here, broad signal in zone 1. Amines. Okay, wait, we have primary. What do we have? We have, I forget what the degrees thing means on that. But yeah, this looks like it. Oh, I think primary. Wait, what is a primary? Yeah, that's what I thought. Okay, so primary amine has this double thing. Okay, so that's definitely it. You have the primary amine. Do you have to explain? Please circle the compound which best fits it. Identify and show the major peaks on the spectrum. Okay, so this is really easy. Okay, so then number 3, we have the amine, but then what's up with the phenol? Do we see the phenol anywhere? Broad signal in zone 1. We don't really have the broad signal in zone 1. But we do have this like characteristic double peak for the primary amine. I'm going to say that this is number 3. The compound 3 is 3. Just because of this. I don't know what all this is. But R.I.P. my granny, she got hit by a bazooka. Okay, let's just do compound 4. So we have an aldehyde. So wait, first off, is carbonyl present? Yes. Uh, let's see. I think we need to actually. Oh, the aldehyde has the double peak though. Oh, wait, maybe that's the. Loki 3 actually might be. I can't tell. Alright, let's follow the algorithm. Is carbonyl present in number 4? Yes, it is. The CO double bond gives rise to a strong absorption. Okay, so first we should have strong peak zone 4, which is 2K to 1600. Then since we have the aldehyde, we should also have 2 signals in zone 2, which is 2800 and 2700. Yeah, maybe. Definitely not this one. I don't know what the hell this is. So first off, let's do the first thing. Zone 4. 2000 to 1600. 2000 to 1600. We have 2 instead of 1. Aldehyde 1725. Okay, so which ones have peaks? Has peaks in zone, has strong in zone 4. I would say 1 does. So 1 does. 2 doesn't. 3 does. 4 doesn't. And 5 does. So these all likely have C double bond O. Okay, then we go to aldehydes should have two signals in zone 2. What are we in now? Zone 4. So zone 2. Okay, so which have two strong signals in 28 and 27. Okay, so that's going to be, let's just do all of them. So, definitely not 1. Definitely not 2. Um, so so 3 does. At so number 3 at 29 and 28. Number 4 is at 29, 50, and 20. Wait, what? Oh, and 29. Okay, then here we have only one peak in zone 2. Okay, so zone, let me just write this down. Zone 1 Zone 3 is from 2500 2000 to 1600. In zone 5 1600 and on. Okay. So I'm going to say that it's basically just 2, 3, and 4 that have peaks in. If you go here, the aldehydes. So what is this? Single bond O? Actually, could we look at that? Single bond O? Because the, okay, so we're looking between number 3 and number 4. So this is at 13. At 15, we have a nitro, but also C double bond O. So maybe that's just like. Maybe that's just the C double, or the not C double bond O, C double bond C.
I think I'm gonna end it. I'm bored. This sucks.